This workflow supports multiple sample types, including cervical, cytology, and mucus samples. Before starting, make sure all required materials are prepared. Additional equipment may be needed depending on the sample type. As the preparation steps may vary, please follow the workflow that matches your sample. To begin, transfer 300 microliters of the sample into the reagent well. Next, add proteinase K to the same well. For mucus samples, start by adding DTT to improve sample processing. Then, briefly vortex the sample to ensure proper mixing. After that, incubate the sample at 37 degrees Celsius for 20 minutes. Once incubation is complete, transfer 320 microliters of the processed sample into the reagent well and add proteinase K to the same well. After preparation, both workflows proceed to automated extraction. Place the consumables into the instrument and start the extraction program. Once the run begins, the system will perform automated HPV DNA extraction.